Let me start out by saying that this presentation will focus primarily on linear growth, and my goal is to provide an overview of the regulation of linear growth. And I admit readily that I will risk oversimplification of the very complex pathways and regulatory systems that are operating to focus really on one main pathway, and that is going to be the growth hormone pathway. So I'd like to also, in this talk, then set the stage for other presentations that you'll hear in the workshop in more detail. So in particular, this audience doesn't need to be told that growth is so important to monitor. It's one of our best overall indicators of child health. It's a very sensitive measure of the environment. It's not highly specific, but virtually every clinic worldwide measures the growth of children, and the growth statistics that are generated often are very useful for large bodies that are monitoring overall health of population. And those growth measures are also very important for tracking the growth of individual children in the context of pediatric care. So I think one of the central themes that I'll be presenting today is that healthy growth reflects harmonization of multiple interacting genetic, hormonal, epigenetic, psychosocial factors that are operating from conception, and I should also say preconception now, to adulthood. And different factors predominate as regulators of growth in different phases. And as I mentioned, a common pathway for the regulation of linear growth involves the growth hormone, insulin-like growth factor, or GH-IFG axis. This is a graphic which I found quite appealing from a very recent publication that shows you the variety of factors that are operating at each phase of growth. And in the preconception and pregnancy periods, we know that there's important roles for genetic variation, for imprinted genes, a wide range of maternal factors, including things like maternal weight, and others that are listed here, as well as some environmental influences. We'll talk more about the individual phases, but I think this gives you an overall sense. It seems like you're going to be seeing a lot of growth curves during this presentation and subsequent presentations, so we'll really be exploring these patterns of growth. And here, growth is depicted as a fairly smooth process that can go awry, but there is potential for catch-up that occurs in childhood and into the pubertal stages. So it's really important to understand in the context of linear growth how bones grow, and in particular to understand the life cycle of chondrocytes, or the cells that are in cartilage and that ultimately get calcified for bone growth. And so in this important growth region at the ends of bones, stem cells are recruited from the resting zone of the bone plate. They replicate in what's called the proliferating zone, and then they increase in the size of cells in the hypertrophic zone. And then mineralization of those cells occurs in the metaphysis of the bone. The progression through these phases is influenced by local and circulating hormone signaling, as well as nutrients. Elongation of the bone stops when the growth plates fuse in the post-puberty period. And often physicians will assess bone age as a way to determine whether a child who perhaps isn't growing so well has future growth potential. Their bone epiphyses have not yet fused, indicating there's still potential for growth. Now I'd like to also just provide a simple view of the central role of the GH-IFG axis. And here we're talking about a very sensitive, delicate regulatory system. The hypothalamus produces releasing hormones that act upon the pituitary, and the pituitary in turn produces growth hormone. Growth hormone has its main effect on the liver, and the liver produces IGF-1. IGF-1 then has a wide variety of influences on other tissues in the body, as you can see in this diagram. And in particular, what we'll be focusing on today is its role in bone growth. But also very important to recognize that it is highly important for muscle growth as well as other organ growth. There's also a complex feedback mechanism operating to keep the hormonal levels in control to optimize the growth of the child. So I'd like to start out giving you a view here of fetal growth. The graph on the left shows you growth velocity in the prenatal period, and we've been able to learn about these aspects of fetal growth by very widespread use now of ultrasonography and fetal biometry. And you'll see that from this graph, estimated fetal weight increases quite dramatically from very early in pregnancy to a peak and then slows down just before birth. These are data from the US National Growth Study, Fetal Growth Study, and they'd show us that there is some race ethnic variation in growth rates, as you can see in the different colored lines, with the highest peak prenatal growth velocity in white populations in the US. The right-hand panels show you other aspects of fetal growth, including the biparietal diameter, but I draw your attention primarily to panel D, which is femur length, and you'll see that there's a rapid acceleration of this growth of a long bone prenatally. It peaks early in the first trimester of pregnancy, and then long bone growth slows down. So this has really important implications, again, for the role of nutrients in this early prenatal period. So in terms of the regulation of growth, there's maternal factors, and in particular, growth hormone derived from the maternal pituitary is operating mainly in early gestation. The mother's liver produces IGF-1 during pregnancy, and those levels of IGF-1 rise across pregnancy. And these maternal hormones that are produced are really very important to regulate the fetal environment, which is so important for fetal growth. The placenta is absolutely central. We can think of it as a communication hub for messages that go from the fetus to the mother. Placental hormones that are produced have both fetal and maternal effects. So, for example, growth hormone that's produced by the placenta regulates maternal metabolic adaptations. For example, the development of insulin resistance in the mom, which will then facilitate the transfer of glucose to the baby. So it's very important that the placenta is regulating not only the growth of the fetus, but also these maternal adaptations. In addition, IGF-1 and IGF-2, another placentally produced hormone, bind to fetal tissue receptors to affect fetal growth. And there's a very important interaction that occurs between maternal nutritional status and placental hormone expression, because we know that IGF-1 is very sensitive to the plane of maternal nutrition, and maternal nutrition can also affect gene expression. And you heard earlier from Andrew that this may occur very, very early on in pregnancy, where the mom's diet has an influence in the early weeks of gestation. Now, the fetus kind of takes over after a bit, and the fetal endocrine system develops by the end of the first trimester, and the fetal pituitary, pancreas, and thyroid start producing hormones that act on various tissues in the fetus to influence growth. Fetal hormones are very important in regulating the growth and organ development directly by affecting nutrient uptake, tissue growth, and maturation of the organs. And we know that insulin is really a primary driver of fetal growth, and it is strongly affected by the amount of glucose crossing the placenta. And as I mentioned before, that control is partly regulated through the placenta. There are other hormones that affect organ maturation, such as cortisol. And I have a few notes on the bottom of some of the slides linking you with some of the presentations that you'll hear later. And in this case, We'll hear more about fetal growth from Dr. Keith Godfrey. So after birth, it's important, and you saw figures somewhat like this in Andrew's presentation, showing you the different phases of postnatal growth. And Dr. Johann Karlberg was well known for advancing what's called the ICP model to represent 3 overlapping phases of growth that are marked by very different growth rates and also by different types of hormonal control. And so during infancy, we see initially rapid growth, and the graph on the side is a velocity curve showing you the rate of growth. So infancy is marked by an initially rapid period of growth and then by decelerating growth velocity. And the main mediators at this point in time are insulin and IGF-1, and they're important mediators of the nutritional status of the child, the developing infant. During childhood, there's a relative plateau in growth rates, a steadier state, and strong control by the growth hormone IGF axis, along with, in addition, the role of thyroid hormones. And at this point in time, there's a strong pulsatile release of growth hormone that can vary in amplitude as well as frequency. Then during puberty, as you'll see in all of these growth charts, there's a rapid growth and maturation. And there's variation in the timing and transitions across these 3 phases that characterize populations, as has been shown by comparing low-income or resource-limited populations with the Swedish populations that were represented by Dr. Karlberg's work. So during infancy, there's a very strong control, as I mentioned before, hypothalamic growth hormone releasing factor and somatostatins, if you remember that figure from the second or third slide, are very important to control growth hormone secretion, and it's very well established in the first year of life. And after about 2 or 3 months, growth hormone becomes absolutely necessary to sustain growth. And typically children who aren't growing well after this time period, who may be experiencing varying degrees of growth restriction, may have growth hormone genetic deficits. Insulin is very important, playing a permissive role in modulating the signaling mechanisms involved with growth hormone. And again, nutrition plays a central role in this period of infancy. In the graph on the right, you see a distance curve at the top and a velocity curve that corresponds to that growth in showing you the growth rates. And you'll see that there's a fairly substantial decline in the rate of growth that occurs across the first 2 years. During childhood, the growth rate is relatively steady at about 6 to 8 centimeters per year on average. And the amplitude of growth hormone pulses actually increase with the age of the child, and the growth hormone pulses are larger in general in males, and they're particularly at night. And females have less day-night variation, diurnal variation, but higher total growth hormone production. Nutrients are very important in this signaling, and in particular amino acids, glucose, fatty acids, are all important for stimulating that release of the growth hormone releasing factor from the hypothalamus. Other key factors that are operating during this time period include thyroid hormones, and thyroid hormones are particularly needed to enhance the growth hormone effect on IGF-1 expression. There's also a direct effect of thyroid hormones on bone cells, the chondrocytes and the osteoblasts. We also know there's a very important role for vitamin D, both in mineralization of bones and also in regulating insulin. Ghrelin is another substance which has received more attention recently. It's produced by the stomach. And some people have suggested that ghrelin levels are good predictors of catch-up growth because of its important role in regulating satiety and hunger. There's been some question about whether or not there's a mid-childhood growth spurt at age 6 and 7, which may co-occur, but is not necessarily regulated by the maturation of the adrenal glands. This has been observed in some populations, but not others. Pubertal growth, as we well know, is characterized by a very large acceleration in height velocity, and this can go up to about 8 to 12 centimeters per year, higher in males than females. And the timing in general of peak height velocity is about 2 years earlier in females versus males. And the main regulation of pubertal growth is the hypothalamic pituitary gonadal axis. So instead of the liver as a target organ, we have the ovaries and testes as the target organs. And the androgens that are produced have both direct and indirect effects on those changes that we see in the chondrocytes in the growth plates. And again, as I mentioned, there's important feedback mechanisms. And in this case, we switch from a negative feedback to a positive feedback, allowing for higher levels of circulating growth-promoting hormones. Growth acceleration is timed differently in different parts of the body, first in the hands and feet, and then in arms and legs. And adolescents at this point in time may seem a bit uncoordinated and gangly because they're disproportionate until all of the growth of the different areas of the body come back into harmony. The timing and intensity of the pubertal growth spurt can be affected by stress and poor nutritional status. And in some cases, the response of the growing child or adolescent is to grow for a longer period of time, to have a less intensive growth spurt with a lower peak velocity. And then, as I mentioned before, linear growth stops with the fusion of the epiphyses. So, as I mentioned, you will see a lot of growth curves, and they depict growth as a process that's very smooth. But is it really? So there's a concept that's called saltation and stasis, and this is exemplified by work by a very good friend of mine, Michelle Lampel, and she demonstrated with very careful daily measurements of infants where the investigators were highly trained to take very accurate measurements, that growth is not continuous, but it occurs in very time-distinct, time-constrained episodes called saltations. And these are followed by periods of stasis or no growth. Saltations are much more frequent in infancy, and as you can see in the graph, it looks like a stepwise function rather than a smooth pattern of growth. And the bottom section of the graph shows you the saltations that are occurring, followed by periods of stasis. So the saltations are actually consistent with episodic gene expression patterns and cyclic bursts of cell proliferation. So there's actually a well-documented basis for this pattern of growth. And in the context of bone growth, variation in the frequency and amplitude of saltations are very important in influencing final height. Nutrient deficits, particularly deficits in amino acids, may decrease the amplitude or totally eliminate the saltations and thus influence bone growth. So while saltations have been observed in several other studies, and including in other places in the world, including the Gambia, there's some investigators that have not observed these saltations, so it's still a bit of a controversial theory. We also know that there's a strong genetic regulation of growth. Growth follows a polygenic inheritance pattern, where classic studies identify that about 80% of variation in stature is explained by genes. And there are genes with roles in the regulation of growth located on all of the chromosomes. The GIANT Consortium recently published a map, saturated map of common genetic variants that explain about 50% of the variation in height, and then suggests that about 3% may reflect their inability to capture all of the SNPs that are involved, or perhaps large effects for multiple rare variants that we have not been able to capture through genome-wide association studies. Much of what we know about genetics of height comes from studies of genetic mutations in laboratory animals involving knockouts or knockdown studies. And the investigations show that there are clusters of genes in different parts of the genome that control skeletal growth. These include things like genes for growth hormone and growth hormone receptor, placental lactogens, etc. So I can't ignore the role of fathers. We know that there are some important effects of sperm epigenetics. Specific methylation patterns in the seminal fluid and in the sperm reflect paternal diet, paternal stress, age, smoking, environmental factors, etc. We also know that microRNAs can carry information about the father's environment, including the father's diet and the father's stress. In addition, fathers play an important role in parental care, and maternal and paternal stress are found to be associated with reduced linear growth. And there was an excellent study done here in Indonesia showing the increased risks of stunting in the absence of paternal care. So I'd like to briefly mention a few of the other factors that are regulating growth. Stress is really very important. Stress is important in the production of glucocorticoids that have direct effects on the pituitary growth hormone production. Stress can also elevate other hormones that can suppress or act against growth hormone. And in addition, the hypothalamic pituitary axis is involved in the conversion of T4 to T3, and there's energy conservation associated with that mechanism, which can also then have an influence on growth. So my last point is going to focus on the idea of nurturing care. Poor growth has been seen in the presence of adequate nutrition, but lack of nurturing care. The term now that we use, rather than failure to thrive, is psychosocial short stature, and this is a well-described condition. So studies on children in institutional settings or following adoption provide very important data. And psychological factors, including emotional deprivation, lack of stimulation, family conflict, and maltreatment, can have persistent effects in elevating stress hormones that are important in those pathways that I've already described to you in terms of growth hormone and IGF-1 regulation, sensitive to the stress that can occur. So there's now a nurturing care framework for child development, and this is a set of guidelines for countries to invest in nurturing care programs to lay the foundation for human capital and a brighter future for children, with the idea that we need not only good nutrition for child development, but also this kind of nurturing care and absence of stress. So there are numerous other environmental factors that you'll hear about in the rest of the talks here: chronic infections, environmental exposures, nutrition during adolescence and the sensitive period there, and the gut microbiome. So you'll learn lots more about the mechanisms regulating growth. So while broadly similar, trajectories of healthy growth vary across individuals and populations. In general, growth is very well canalized with genetic constraints that set the boundaries for growth, and then many factors influencing compensatory growth, either a slowing down or a speeding up of growth, operating through nutritional and regulatory pathways. Hormonal and physiologic regulation of growth is an amazingly fine-tuned and very complex process. So I'd just like to end with a key message, which is that healthy growth reflects the harmonization of multiple interacting genetic, hormonal, nutritional, epigenetic, and psychosocial factors. Thank you very much.